Aliana Castronevo and I'm here to discuss an overview of protein translation. So to begin with, let's take a look at our hand. Our hand is composed of the nails, the muscles, the bones, and the skin, and all of these are composed of cells. And each cell contains millions and millions and millions of protein. As a matter of fact, proteins are what we call the key molecular building blocks of every organism living here on Earth. So the question is, how are these proteins made into cell? For starters, the instructions for making proteins are written in a cell's DNA in the form of genes. If you are kinda new to this process, you might want to check out the central dogma for the DNA to RNA to protein process before getting into this kinda nitty-gritty process. So basically, a gene is used to build proteins in a two-way process. The first process being the transcription. A transcription is a stage wherein there is a production of the mRNA or the messenger RNA. The second one is the translation. The translation is a process wherein the mRNA or the messenger RNA is being decoded to build new proteins or even chunk or subunits of proteins. So, to understand more about this translation, let me ask you this question. Have you ever wondered how antibiotics kill a bacteria? For instance, when we have that very annoying sinus infection, different antibiotics works in different ways. But some antibiotics attack a very basic process in a bacterial cell, and that is, they knock out the ability to make new proteins. To use a little more molecular biology vocab, an antibiotic blocks translation. In a process of translation, there is this cell called the messenger RNA that transmits information, and it uses this information to build new proteins. Did you know that the translation happens every day in all bacterial cells, as it is into all cells in our body? Actually, it is the key of keeping us alive and also keeping our bacterial visitors alive. When you take a certain antibiotic molecule, this antibiotic molecule will latch on a key translation molecule inside a bacterial cell and basically stall them with no way to make proteins. So then the bacteria will stop functioning and then it will eventually die. That's why infections clear up when they're treated with antibiotics.
Hi, I'm Trisha Calma. So, the mechanics of protein synthesis, we have three major steps, and these are the initiation, the elongation, and the termination. In the initiation is the assembly of the initiation complex, while in the elongation is the adding of new amino acids on the sequence one by one, and the termination is the release of the completed polypeptide and dismantling of the ribosomal machine. Translation is the process whereby the base sequence of the messenger RNA is converted to an amino acid sequence of a polypeptide. This involves an mRNA, tRNAs, amino acid, and a ribosome and other accessory proteins. The mRNA consists of three main parts. The portion of the 5' end to the translation start codon is the 5' untranslated leader region, more simply called the leader. The central portion is called the reading frame. It commences with the start codon UAG followed by a series of 3 nucleotide codons, each of which specifies an amino acid, and it ends with one of the stop codons UAA, UAG, or UGA. The final portion of the mRNA consists of the section from the stop codon to the 3' end called the 3' untranslated trailer sequence, more simply called the trailer. mRNAs vary in length. because of differences in all three parts of the molecule. Transfer RNA functions to bring amino acids to the growing polypeptide during translation. It consists of a single RNA chain about 75 to 90 nucleotides, each tRNA having a different sequence of bases. After it is transcribed, the tRNA folds up into a characteristic cloverleaf structure. The anticodon contains of 3 nucleotide sequence that is complementary to a codon on the mRNA. The amino acid is attached to the 3' free end at the top of the clover. The ribosome assembles amino acids into polypeptide. Ribosomes consist of two parts, a large subunit and a small subunit. Ribosomes are made of a ribosomal RNA and various proteins. The active ribosome contains 3 tRNA binding sites, namely the E, exit, P, the peptide, and A, the amino acyl. In prokaryotes, translation initiation occurs in 3 steps as follows. First, the small ribosomal subunit complexes with 3 proteins named the initiation factor 1, initiation factor 2, and the initiation factor 3. A molecule of GTP also attaches. The ribosome IFGTP complex recognizes a sequence in the leader upstream of the AUG start codon. This sequence is called the ribosome binding site or the Schrein-Delgarno sequence after its discoverers. The sequence is complementary to a sequence at the 3' end of the 16S ribosomal RNA of the 30S ribosomal subunit and serves to align the ribosome on the mRNA in its proper reading frame. Second, translation begins at the start codon, AUG, a few bases downstream of the ribosome binding site. The first AUG in protein specifies a modified form of methionine. This modification consists of the addition of formyl group to methionine, making it formyl methionine. The anticodon of this initiator tRNA containing FMET complements AUG. Upon binding of the FMET tRNA of the mRNA, the IF3 is released with the remaining assembled components called the 30S initiation complex. Lastly, the large ribosomal subunit binds to the 30S initiation complex. GTP is hydrolyzed and IF1 and IF2 are released. The final complex is called the 70S initiation complex. The FMET tRNA is now situated in the P site of the ribosome with its anticodon hydrogen bonded to the mRNA start codon. Initiation is now complete and elongation follows.
Hi, I'm Kate Lohica. So we now know that protein synthesis is a 3-step process including initiation, which was explained earlier, elongation, and termination. We'll now focus on the elongation. Elongation is the adding of new amino acids on one by one. It involves repetition of 3 steps. First, an elongation factor called EFTU, which is associated with GTP. This EFTU binds free charged amino acid tRNAs or the transfer RNAs. This complex enters the A or the acceptor site. Correct selection of the tRNA complex depends mainly on the codon-anticodon pairing in this example. The anticodon CCC pairs with a GGG codon. The tRNA carries the amino acid. In the second step, the ribosome's peptidyltransferase activity catalyzes the formation of a peptide bond between the new amino acid in the A site and the previous amino acid in the P site. Simultaneously, GTP is hydrolyzed and the resulting EFT-GDP is expelled. The third and last step is called translocation. An elongation factor called EFG, it's also associated with GTP, binds to the ribosome. The GTP is hydrolyzed, providing the energy to RAShe. The 50S and 30S submits a head 1 codon. This maneuver opens up the A site and slides the uncharged tRNA to the last side called E for exit. The next amino acid tRNA that enters the A site creates a conformational change in the ribosomal and telegraphs through to the E site and ejects the uncharged tRNA. These elongation steps repeat along the mRNA. The ribosomes of E. coli can speed through these elongation steps, linking together amino acids per second. Eventually, the ribosome arrives at the end of the coding region, marked one by one of the stop codons. This stage of translation is called termination, and it will be further discussed by our next speaker.
Hi, I am Claudia Regala and I will discuss the termination stage. In the video, you will notice that we have reached a codon, the UGA, which is one of our 3 termination codons, without a corresponding amino acid. This is a signal to the ribosome that it is done translating this amino acid or this messenger RNA strand, which means that we're in step 3, the termination stage. Now, the components of the initiation complex, the large and the small subunits, are going to disengage and release the amino acid sequence that will go on and take its final 3-dimensional shape. Now, in this case, we have a very short amino acid of only 4 amino acids. This is a very small protein, but imagine this to be 100 or 200 amino acids long. And we are seeing it a very linear way. But we know that proteins will twist, fold, and kink into a complex 3-dimensional shapes as it takes on its secondary and tertiary structure based on hydrogen bonding interaction and R group interaction. Now, these 2 ribosome components can go on to reform or maybe go back to the beginning and start to translate this messenger RNA codon a second time to make another copy of this protein. And this messenger RNA strand can be translated many times by many ribosomes. The orange line down here represents our messenger RNA strand, and the ribosomes are moving down like trains on a track, one after the other, moving from left to the right. A very good example is shown here. We can see the messenger RNA strand, and the dark spots that are along it are the individual ribosomes that are traveling down the track. And you can see the proteins are kind of coiling off of the ribosomes. As they are being constructed, as the ribosomes move down the track here. So the question is, why does the mRNA need a stop codon? When a ribosome reaches the codon UAG, UAA, or the UGA on an mRNA strand, then protein synthesis is terminated. Since a stop codon does not have a corresponding tRNA, protein known as a release factor is released instead. It then binds to the ribosome and adds a water molecule to the end of the amino acid chain. This reaction hydrolyzes the polypeptide chain from the tRNA, allowing the protein to exit the ribosome.
Hi, I'm Cedric Makatantan. So what else do we know about protein translation? Translation is the process by which the base sequence of an mRNA is used to order and to join the amino acids in a protein. The 3 types of RNA participate in this essential protein synthesizing pathway in all cells. In fact, the development of the 3 distinct functions of RNA was probably the molecular key to the origin of life. For the RNA molecules that can be seen and their functions, there are the messenger RNA, the transfer RNA, and the ribosomal RNA. Messenger RNA carries the genetic information copied from DNA in the form of a series of 3 base code words each of which specifies a particular amino acid. So transfer RNA is the key to deciphering the code words in mRNA. Each type of amino acid has its own type of RNA, which binds it and carries it to the growing end of a polypeptide chain. If the next code word on mRNA calls for it, the correct tRNA with its attached amino acid is selected at each step because each specific tRNA molecule contains a 3-base sequence that can base pair with its complementary code word in the mRNA. So ribosomal RNA associates with a set of proteins to form ribosomes. These complex structures, which physically move along an mRNA molecule, catalyze the assembly of amino acids into protein chains. They also bind tRNAs and various accessory molecules necessary for protein synthesis. Ribosomes are composed of a large and small subunit, each of which contains its own tRNA molecules or molecules. So how does protein translation work? Translation is the conversion of information contained in a sequence of mRNA nucleotides into a sequence of amino acids that bond together to create a protein. The mRNA moves the ribosomes and is read by tRNA, which analyzes sections of 3 adjoining nucleotide sequences called codons on the mRNA and brings the corresponding amino acid for assembly into the growing polypeptide chain. The 3 nucleotides in the codon are specific for a particular amino acid. Therefore, each codon signals for the inclusion of a specific amino acid, which combines in the correct sequence to create the specific protein that the DNA coded for. The assembly of the polypeptide begins when a ribosome attaches the start codon located on the mRNA. Then, tRNA carries the amino acid to the ribosomes, which are made of RNA and protein and have the 3 bonding sites to promote that synthesis. The first site orients the mRNA so that the codons are accessible to the tRNA, which occupy the remaining 2 sites as they deposit their amino acids and then release from the mRNA to search for more amino acids. Translation continues until the ribosome recognizes a codon that signals the end of the amino acid sequence. The polypeptide, when is in its primary structure, it is then released from the ribosome to begin contortions to configure it to the final form to begin its function. Transfer RNA plays the role of carrying an amino acid to the synthesis site at the ribosome. They form several loops, one of which is an anticodon, a 3-residue series that is complementary to the codon present in the mRNA, which can be seen in the figure. The opposite end of the tRNA is where an amino acid is bound. So for the overview of the 3 processes, with the initial methionine in place, another tRNA with its amino acid joins the assembly site as dictated by the codon on the mRNA. With 2 amino acids present, a peptide bond can be formed and the polypeptide can begin forming. The new amino acid is added to the carbon end of the polypeptide with the peptide bond forming between the CO of the polypeptide and the amine of the new amino acid. This structural specificity is enforced by the nature of the binding between the amino acid and the tRNA. The portion of the amino acid that is unbound in the tRNA complex is the amine. So elongation ultimately requires the repetitions of several steps, as can be seen here. So the structure of the ribosome plays an important role in this elongation process. There must be 2 sites available for synthesis to occur. One site, called the P site for peptide, is where the growling or nascent polypeptide is located. Adjacent to this location is another site where the tRNA with its new amino acid can bind. This site is called the A site for the amino acid that is delivered there along with the tRNA. So why is protein translation important? It should be noted that we need protein in our lives, given that protein synthesis is extremely important to life in general, and being a step in protein synthesis, protein translation would also be of great need to carry out the synthesis. Almost everything that carries out a cell's functions is regulated by proteins. Protein translation is important because the proteins created during this process control the activities of the cells. Without these proteins, many of the processes in the body would fail or not work properly. Protein is key to such reactions as metabolism, DNA replication, and molecule transportation. They are also key in making sure the human body reacts properly to stimuli. If the human body did not get enough protein, then it would not be able to replicate DNA, which means that the body would not be able to heal itself, reproduce, or grow as needed. Each protein has a unique amino acid sequence which is given to it by the gene that encodes it. These sequences help determine what the protein will be used for inside the body. The protein that is not ingested through diet is synthesized through the process of translation. So where in the cell does protein translation take place? In a eukaryotic cell, the final stage of protein synthesis, which is the translation, takes place in the cytoplasm after the initial stage, which happens in the nucleus. In a prokaryotic cell, there is no need for the second stage of the process. Translation takes place in the cell's cytoplasm. During this stage, genetic information is transferred from RNA to protein. Peptide bonds between amino acids are formed and thus protein is synthesized. The ribosomes in the cytoplasm are the sites of protein translation in the cell. Translation refers to taking a copy of messenger RNA and making a protein from the code. At the start of translation, the ribosomal subunits assemble to form the ribosome. The mRNA is read or translated 3 codons at a time. Each set of 3 codons stand for a certain amino acid. Transfer RNA bring their amino acid loads to add to the growing protein chain.